All right, so a quick reminder of what we've discussed in our last session. We stated that we have 4 different types of tissues that are going to be forming all the body organs. If you remember, those are the epithelium, connective tissue, and muscle and nervous tissue. Those are the 4 different types of tissues that we have forming together all the body organs. We did discuss last class the epithelium, and if you remember, epithelium was classified according to 2 factors. First one is according to the number of cell layers. If I have only one row of cells forming the epithelium, this was classified as a simple epithelium. If I did have more than one cell layer forming my epithelium, we classified this as stratified. The other factor according to which we classified the epithelium is according to the shape of apical cells. So if I have flat cells at the apex forming my free border, we call those are squamous cells, and this would be classified as a squamous epithelium. If the apical cell layer was formed of cube-shaped cells, we call this as a cuboidal epithelium. And if the apical cells are tall cells, we classify this as a columnar epithelium. So what are the different epithelial types that we've seen last class? If you remember, we first have seen an epithelium that's formed of one cell layer, and those cells were squamous, so we classify this epithelium as a simple squamous epithelium. We then have seen a type of epithelium which cells are going to be forming just one layer, and the shape of those cells were cuboidal. They were cuboidal cells, so we classified this as a simple cuboidal epithelium. Seen another type of epithelium in which cells are forming one layer, and they are tall cells, they are columnar cells. We classify this as a simple columnar epithelium. And something to remember when you are mentioning simple columnar epithelium, you need to remember that we have 2 varieties of the simple columnar epithelium. We had simple columnar, which cells had hair-like projections coming out of their apical surface. We called this a ciliated simple columnar epithelium. And we did have another variety of the simple columnar epithelium, which cells didn't have any hair-like projections or cilia. So we call this one is a non-ciliated simple columnar epithelium. We also have seen last class a type of epithelium which is formed of more than one cell layer as it appears. But when we looked at the shape of the apical cells, we find out that those are tooled cells or columnar cells. So for the first impression, when I looked at this tissue, I've seen that I have more than one cell layer, but when I look at the apical cells, the cells at the apex of the tissue, the top cell layer, we've seen that the cells are columnar. And remember, we mentioned that this is not something logical. Why? Because if I was formed of multiple cell layers, the apical cells are the ones which are going to be located away from the basement membrane. They are located away from the basement membrane. So if you remember, the epithelium is avascular. I don't have blood vessels. And I need to be supported by the connective tissues that allows the diffusion of fluid carrying the oxygen and nutrients. So as I keep moving away from the basement membrane, I'm expecting the cells at the top cell layer to receive less oxygen and less nutrients. So this means they were supposed to be shrinking. When I check here, I see that the apical cells are columnar, so it's not something logical for me. So we looked closer and we figured out that all the cells are attached to the basement membrane. So if all the cells are attached to the basement membrane, this means I'm only formed of one cell layer. But one cell layer that gives me the impression, a false impression, because the cells are so crowded that I'm looking at multiple cell layers. And what gave me this impression is that the nuclei of those cells are arranged on different levels. So I get a false impression that this is a stratified, although it's simple. It's only formed of one cell layer. All the cells are attached to the basement membrane. So what did we classify this epithelium, which is simple, but gives me the impression that I'm looking at a stratified epithelium. We called it a false stratified, pseudo stratified. And what is the shape again of the apical cells? The cells at the top cell layer. It's columnar, so we're gonna call this epithelium, it's my pseudo-stratified columnar epithelium. But remember, this is a simple columnar, but the cells are so crowded. So it's also gonna have the same two varieties as a simple columnar epithelium. So in some cases, the cells will have those hair-like projections coming out. And in some other cases, the cells don't have those hair-like projections coming out. So what did we classify those two varieties of the pseudo-stratified columnar epithelium. We classified this as a ciliated simple or pseudo-stratified columnar ciliated or columnar epithelium. And this one was the non-ciliated pseudo-stratified columnar epithelium. We also have seen another type of epithelium in which I've seen many cell layers. Cells at the base are much longer, they are much larger than the ones that are going to be closer to the surface of the epithelium. And when I looked at the apical cells, I figured out that this was formed of flattened cells. So what did we classify this epithelium? We classified the epithelium here as it is formed of more than one cell layer as a stratified, and as the apical cells are flat cells, this is going to be classified as a squamous epithelium. So this is going to be stratified squamous epithelium. But remember, when you are about to mention stratified squamous epithelium, you need to remember that we do have two varieties for the stratified squamous epithelium. The first variety did have keratin layer on top of my squamous cells. And how did I know that this is a keratin layer, not another layer of cells? Remember, living cells do have nuclei. So all those cells will have nuclei. Here, those are fibers, just fibers on top of the living cells. So we classified this type of epithelium as keratinized stratified squamous epithelium. And the other variety was formed by the multiple cell layers, and the top cell layer was formed of flat cells. But I didn't have this keratin layer on top of my squamous cells, so we classified this one as a non-keratinized stratified squamous epithelium. The last type of epithelial tissues that we've seen last class was a type of epithelium formed of multiple cell layers. Some of the apical cells were flat and some others were cuboidal. You see here some of the apical cells were cuboidal cells and others were squamous cells. And remember, we call this type of epithelium because it has the ability to change the shape of its cells to allow stretch to take place. If you remember, this was my exactly transitional epithelium. All right, so where did we get each of those types of epithelium? First, starting with the simple squamous epithelium. Remember, simple squamous epithelium, I'm formed of one cell layer. Those are flat cells. I need to have a minimal resistance. Where do we need to have a minimal resistance? Exactly, Theodore, alveoli. What part of the nephron did need to allow the filtration to take place? So you're going to need a very low resistance, not the whole nephron, but a specific part of the nephron we've called the glomerulus of the nephron. Glomeruli, exactly. Where else did we need a minimal resistance? In the blood vessels, in the chambers of the heart. So lining the chambers of the heart, lining the blood vessels. I need to have a minimal friction against the blood flow. So those are going to be aligned with one row of flat cells. Simple cuboidal epithelium, remember this is a type of epithelium that allows absorption and secretion, and we find this type of epithelium in the kidney tubules. And the kidney tubules are part of my nephrons. That's why I was asking here, what part of the nephron? Because the kidney tubules are actually part of the nephrons. So I need to be more specific. I can't just say kidney. Kidney will have different types of tissues, but I need to say kidney tubules. And those are the tubules that will be allowing the absorption and secretion, allowing you to modify the structure of the filtrate to form the urine. All right, you see here the glomeruli of the kidney, the glomeruli of the nephrons, those are parts of the kidney. Kidney tubules are part of the nephron, so I can't just keep it general. All right, so I need to be more specific when I state the location of a specific tissue. Simple columnar epithelium. I had ciliated and I had non-ciliated. If you remember, where did I have ciliated, which is going to be responsible for secretion of mucus, secretion of enzymes, allowing the absorption. Remember, this was located in the gastrointestinal tract, all the gastrointestinal tract except for the oral cavity, pharynx, and esophagus. So starting from the stomach all the way to the anal canal, and this includes stomach, small intestine, and large intestine. All this part is lined with simple columnar epithelium, which is non-ciliated. How come? Some people get confused in here. How come you are able to push the food throughout the gastrointestinal tract without having cilia? So some of you get confused on this. This is a very common wrong answer I see on the exams. How come am I able to move the food down the gastrointestinal tract without having cilia? Because you rely on muscular contraction, not the propulsion of the cilia. So here I don't need cilia because I have major contractions taking place. I have peristalsis, exactly. So here, I don't need cilia to allow the food to move, although some of you will think I should have cilia in the gastrointestinal tract. And this is what gets many of you confused on the exam. We do not have cilia in the gastrointestinal tract. All right, so the type of epithelium is a non-ciliated simple columnar epithelium. How about the cilia? Where would I actually need? I don't have much contractions, and I will need to allow the propulsion of mucus by having ciliated simple columnar epithelium. Remember, in the uterus, parts of the uterus and the uterine tube. The uterine tube is the site for normal fertilization of the ovulated egg. So fertilization of the egg takes place in the tube, unlike what most of you think that it takes place in the uterus, in the cavity of the uterus. No, it takes place in here, in the uterine tube. But this growing morula zygote is gonna need to be transported for it to be implanted in the endometrial cavity, because remember, the wall of the fallopian tube or the uterine tube, it's not gonna be able to accommodate this baby. So you need to move the fertilized egg from the tubes to the endometrial cavity. And what allows me to move the fertilized egg, again, the presence of simple columnar ciliated epithelium lining the fallopian tubes. What else? If you remember, we mentioned that in the airways, the small bronchioles were lined with simple columnar ciliated epithelium. And again, why do I have simple columnar ciliated epithelium? Because you are always breathing dust particles, and you don't want to accumulate those dust particles in your lungs. So what you do, you're gonna be releasing mucus, and those dust particles will stick to the mucus. And as they accumulate all the time, you're gonna be pushing them up towards the trachea. And we're gonna see in the next type of epithelium what is lining the trachea. So the trachea is not going to be lined with simple columnar ciliated epithelium. It's going to be lined with pseudo-stratified columnar ciliated. This one on here, this is the one that will be lining the trachea, which is pretty similar to the simple columnar ciliated. It's the only difference between those two types is that here the cells are crowded, giving me an impression I'm looking at more than one cell layer. But they serve the same purpose, which is the secretion of mucus and the propulsion of this mucus. But here in the bronchi of the bronchial tree of the airways, they are simple columnar compared to in the trachea, you're going to have more crowded cells forming the epithelium. So this is going to be pseudo-stratified columnar. But keep in mind, both of them are releasing mucus. Both of them will have cilia to allow the propulsion of the mucus to the outside of your airways. And again, what you're going to do with this mucus that you have pushed up all the time, you're going to be swallowing this. It's a very thin film. So only the columnar cells you see on here, the two examples of ciliated epithelium are either ciliated columnar, simple columnar, or ciliated pseudostratified. All right, those are the two major ones. All right, so again, what will I do with this mucus? It's gonna be swallowed and digested. What happens if a person was a smoker? Remember, the smoke of the cigarettes will be inhibiting the propulsion of the cilia. And as I inhibit the propulsion of the cilia, what happens? I fail to push this mucus out to clear my airways. And this mucus will start to accumulate, forming plugs. It's going to be plugging my airways. So you will see a smoker person, he's going to have more tendency to cough. Why would I be coughing? Because of the accumulated mucus in the airways. The bronchi and the bronchioles will be irritated, and the cilia fail to push this thick and dense mucus. So as they get irritated, you start to create a positive pressure within your chest, trying to dislodge this mucus plugs. Seen here, pseudo-stratified columnar, either ciliated or non-ciliated. Remember, they serve the same purpose as a simple columnar, ciliated and non-ciliated. Pseudo-stratified columnar ciliated epithelium. This was the type of epithelial lining the trachea. And the non-ciliated type is not common. It's mainly gonna be in the sperm carrying duct in the male reproductive system. So those ducts that are transporting the sperms from the testis, in some cases, they will be aligned with non-ciliated simple columnar, non-ciliated pseudostratified columnar epithelium. Moving on to the stratified squamous epithelium. Remember, the cells are kept growing, dividing, and replicating, traveling upwards to the extent where they shrink to become squamous, or even in the keratinized, those cells, as they keep moving away from the basement membranes, they get much less oxygen and nutrients. So what's going to happen to them? They will die. So those are dead cells. Those are the remnants of dead cells that form the keratin layer. So why would I grow the number of cell layers? What is the logic in here? What do you expect to be the function of this type of epithelium? If I'm telling you that this type of epithelium is going to be increasing the number of its cells to reach the maximum barrier for protection. Exactly. So I'm creating a barrier that is responsible to protect, providing me with a high level of protection to the underlying tissues. So which one gonna be providing you with more protection, keratinized or non-keratinized? The one that has extra layer of fibers, or the one without the extra layer? Of course, it's gonna be my keratinized. So keratinized stratified squamous epithelium is the one that provides me with a maximum protection compared to the non-keratinized. It's also providing me protection, but not the maximum protection like the keratinized. All right, so where do I need the maximal protection? Where do I need the maximal protection? In the part of the body that is exposed to the outside. So everything outside of the body is getting in contact with what part of your body? With your skin. So the outermost layer of the skin is gonna contain keratinized stratified squamous epithelium. What is the outermost? The top layer of the skin, or located on top of the skin. So this is gonna be on top of is epi. And when you go to the skin doctor, you go to a dermatologist. Derma skin. All right, so on top of the derma, so we call this is my epidermis, because we're gonna see other types of tissues that are sharing in the formation of the skin. So remember to be more precise in your answers. All right, not just skin, it's gonna be in the epidermis of the skin, because we're gonna have many layers, and each of the layers of the skin is gonna be formed by a different type of tissues. Remember, this is a whole concept. An organ is formed of different types of tissues, not only one type of tissue. All right, so the epidermis, this outermost layer of the skin, is formed of keratinized stratified squamous epithelium. How about the lining that requires high level of protection, but not the maximal protection? Remember, those are parts of the body exposed to the outside, but they are not exposed all the time to the outside, like the skin. So this is going to be like the lining of the oral cavity, the pharynx, and the esophagus and vagina. Those are parts that are exposed from time to time to the outside. So you see on here, that's why I can't say here the gastrointestinal tract is lined with simple columnar non-ciliated epithelium, because part of the gastrointestinal tract, which is the oral cavity, pharynx, and esophagus, those are not lined with non-ciliated simple columnar. They are lined with non-keratinized. Stratified squamous epithelium. All right, so that's why if I was asking a true and false question, for example, so I would ask you, non-ciliated simple columnar epithelium is lining all the gastrointestinal tract. Is this going to be a true or a false statement? What do you think? False, right? False. Why is it false? Because oral cavity, pharynx, and the esophagus are part of the gastrointestinal tract, and they are not lined with simple columnar non-ciliated epithelium. They are lined with non-keratinized. And remember, those are the parts of the gastrointestinal tract that get first in contact with anything that you ingest. So when you ingest food, the food will get in contact with the oral cavity first, pharynx, and then esophagus. So those are exposed directly to the outside. Moving on to the last type of epithelium that we discussed in our last session. This was the transitional epithelium. And remember, transitional epithelium, when I looked first at, I decided to say stratified, because I find out that it's formed of more than one cell layer. And when I look at the apical cells, this is what gets me confused. Some cells were flat, some cells were cube-shaped cells. So I wasn't sure whether it's squamous or cuboidal. So I decided to not consider this way of classification. I will be considering the fact that this epithelium has the ability to change the shape of its cells. So we called it transitional epithelium. And remember, why do I have transitional epithelium? To allow stretch. And in what part of your body do you need to allow stretch? What parts are going to be aligned with transitional epithelium? Yeah. So most of you did answer the most common wrong answer. Most of you did answer the most common wrong answer, which is uterus. Transitional epithelium, it's not located in the uterus, is not lining the uterus. All right, I'm not sure why people keep answering uterus. It's not only you, so I've seen this for over than 6 years teaching this class. The most common wrong answer for the question asking about the location of the transitional epithelium, I got uterus as an answer. So I have two explanations why people say uterus. Tell me if I'm right. So uterus, because some people will get just confused on how to spell. So they know that it's the ureters, which are the muscular tubes that will be conducting the urine down from the kidney to the urinary bladder, or you get just confused with urethra, also looks similar to uterus or ureters. All right. This is one explanation. Another explanation is that when you think of it, you think that it's logical because during pregnancy, the uterus is going to be expanding, right, to accommodate the baby. So you're going to have stretch of the uterus. So you see here, this is a type of tissues that allows stretch, and it is so logical that I get stretch in the uterus during pregnancy. So uterus is lined with transitional epithelium, but please pay attention here that the uterus does not expand overnight. It does not expand overnight. It takes 9 months for it to stretch. It doesn't stretch overnight. Compared to the urinary bladder, how many times do I stretch the urinary bladder a day? 3, 4 times. So each time you stretch and recoil, stretch, recoil, stretch, recoil. So you need something that allows stretch and recoil, stretch, recoil, stretch, recoil. But here, I've got a lot of time to build up new cells lining the endometrium. I don't want to have cells that can stretch here. I've got a lot of time to build up new cells. So I'm building up a whole lining, increasing the thickness of the endometrium of the uterus because I got a lot of time here. But in the urinary bladder, in the ureter, in the urethra, you expand them multiple times a day. So you need a type of epithelium that it allows you to expand and recoil, expands and recoils, expands and recoils. All right. Esophagus. So for some of you are asking about where did the non-keratinized stratified squamous epithelium was located. It was lining the oral cavity, pharynx, esophagus, and vagina. All right, so again, where is the transitional epithelium located? Not the uterus. All right. Where is the transitional epithelium is located? It's located in the urinary tract, ureters, urinary bladder, and urethra. Not the uterus, not the uterus. All right. This is briefly what we have covered in our previous session. All right, so remember, epithelium was forming the linings and coverings, as this is what we discussed last class and earlier today. But another type of epithelium is the glandular epithelium, and this part does not play an important role for our lab exam. So what we're gonna do for this part, the glandular epithelium, the classification of the glands, which are also formed of epithelial cells, we're gonna skip this part because it doesn't serve us for the lab portion. We're gonna get back to it at the end of the chapter. So when you are preparing for your lab exam, remember that glandular epithelium, it's not gonna be tested in the lab portion of this chapter. All right, so our first lab exam is not gonna be covering the glandular epithelium. All right, so that's why we're gonna skip it for now, and we're gonna go back to it to cover it after we complete this chapter. Moving on to our second type of tissues. Remember, we've got 4 types of tissues: epithelium, connective tissue, muscle, and nervous tissue. So today we are starting our discussion of the connective tissue. And the connective tissue here, if you remember, was responsible to form the internal structures and to connect, to attach the different tissues to one another. We have 4 subtypes of connective tissues. We have the connective tissue proper. We have cartilage. We have bone. And we have blood. So all those are considered types of connective tissue. So again, 4 types of connective tissues that we're gonna be discussing. We have proper connective tissue or connective tissue proper, cartilage, bone, and blood. Another name here for the bone tissue is osseous tissue. It's the same as bone tissue. Generally speaking, a connective tissue has 3 major components. It has a matrix, the fluid in which you keep cells and you keep fibers. So looking at connective tissue, we have three major components of any connective tissue. We have cells, fibers, and those are embedded in a matrix. Three components on here. We're going to see different types of cells according to the type of connective tissue we're going to be looking at. One of the most important things that you need to consider are the three major types of fibers, because this will guide you to identify the type of connective tissues that you're looking at. So three types of connective tissue fibers. First one, it's formed of thick bundles like this. Thick bundles of fibers. This is called the collagen fibers. So I have thick fibers, and those are my collagen fibers. I have thin, darkly stained. Here is gonna be light colored because I have a like a dark background. Thin, darkly stained long fibers. Those are my elastic fibers. A third type of connective tissue fibers, those are going to be short and branching fibers. Those are called my reticular fibers. So again, how many types of fibers should we focus on in order to identify the type of connective tissues that we're going to be looking at? Three types of connective tissue fibers. First, we have collagen fibers, which are thick bundles. We have branching fibers, those are my reticular fibers. And we have thin, darkly stained long fibers, those are my elastic fibers. So let's check on here if you will be able to identify the different types of fibers. What do you think? What do you think is this one? All right, so please answer this as your first question for today's in-class activity. So what do you think is this type of fiber? Does it look like a collagen fiber, a reticular fiber, or an elastic fiber? Collagen, right? So please, on your in-class activity sheet, please put number 1 and collagen as an answer. So yes, it's collagen. How about this one? See, this thin, darkly stained long fiber. This is number 2 on here. What do you think? Elastic, exactly, elastic. About this one? See this fiber? Please answer this as number 3 for today's in-class activity. Is this a thick bundle of fibers? Is it thin, darkly stained long fiber, elastic, or is it branching, reticular? Reticular, exactly, reticular. Reticular fibers. What's this one? Please answer this as number 4 for the in-class activity. What do you think? Are those thick bundles of fibers or thin, darkly stained long fibers, or they are branching fibers? What do you think? Collagen fibers, exactly. Collagen fibers. Collagen fibers. Let's see this one. What do you think? Answer this as number 5 here. What do you think are those fibers? See on here. So all this is a fiber, you see, the whole thickness. Elastic. Which one do I look like in here? Do I look like collagen? Compare it to this diagram. Do I look like collagen or a thin, darkly stained long fiber or a branching fiber? How would I know if it's thick or thin? If I kept increasing the magnification power of the microscope, everything that is thin is going to appear thick. How would I know? You need something that will also be magnified in the same strength. So you need to compare it to the size of a cell. If my diameter is greater than the diameter of the cell, so this is a thick bundle. If the diameter is smaller than the diameter of a cell on the same slide, it's going to be a thin. So how about this one? Are those fibers have a thickness of diameter larger or smaller than the diameter of the cells in the same slide? What do you think? So this is a cell here. This is the diameter of a cell. So a collagen fiber has a larger diameter compared to the elastic fiber has a much smaller diameter. So those are going to be what fibers? Knowing this. Those are going to be collagen fibers. Collagen fibers. Yes. So number 5 here is going to be collagen fibers. How about those ones? What do you think? This is number 6 on here. So identify the type of fiber. Thin, darkly stained, long fibers. Those are my elastic fibers. Elastic fibers. Let's see this one. What do you think about those fibers? Around the cells. Remember how the reticular fibers did look like? They have a thick diameter compared to the cell. So I do have a thick diameter. I'm not a thin fiber like the elastic. See on here? How about this one? If we're looking here, fibers are, you see, this is the diameter of the fiber. This is the diameter of the cell. So what do you think? Is this a small diameter or a large diameter? Very, very small diameter. So it's a very thin type of fibers. Very thin type of fibers. So this is going to be number 7 for today's in-class activity. This is going to be what kind of fibers that has a very thin diameter, which is darkly stained and long. This is going to be my, waiting for some answers. I've seen a couple of you did answer this. So yes, elastic fibers. Elastic fibers. How about this one? Fibers. And those fibers are in bundles, if you see on here. And this is the diameter of the cell, a nearby cell. What do you think? Collagen. All right, so answer this as number 8. I have thick bundles of fibers. Thick bundles compared to what? Compared to the diameter of the cell. See, all this is a bundle. All this together is a bundle of fibers. And embedded in between is going to be the cells. So those are thick bundles of fibers which have a very large diameter compared to the diameter of the nearby cells. This is going to be, those going to be my collagen fibers. All right. Any troubles identifying the different types of fibers? We're gonna rely on this information to identify the type of connective tissues that we're gonna be looking at. This is very, very, very important to be able to identify the different types of connective tissue fibers. Do you have any trouble identifying those 3 types of connective tissue fibers? Collagen fibers, elastic fibers, or reticular fibers? Bundles of fibers which are thick, they have a wide, large diameter, those are collagen fibers. Thin, darkly stained, long fibers, those going to be elastic fibers. And the ones with branches, those going to be reticular fibers. Exactly. So you need a lot of practice in order to master this. All right, your eye needs to get used to looking at those tissues in order for you to figure them out. This is very, very important after today's class to start, for you to start completing your visible body courseware lab assignment, because this will give you a lot of pictures different than those ones that you have in your textbook, for you to be able to practice and to get used to the appearance of the tissue under the microscope. All right. Any questions? Any questions? We can have a break for 10. Any questions before we go for a break?